Namaskar दोस्तों, welcome to channel Toxicology on Board. Today in this video we'll be discussing another important topic, DABT topic, that is botulism toxin. So let's directly dive into the question. The question is, botulism toxin inhibits neuromuscular communication by option A, blocking sodium channels along the motor nerve, option B, inhibiting acetylcholine release from the axon terminal, option C, inhibiting the reuptake of choline resulting in acetylcholine depletion, option D is evoking unstimulated acetylcholine release. The right answer will be in the green text, that is option B, inhibiting acetylcholine release from the axon terminal. Okay, what is happening in the case of botulism toxin is, this is your neuromuscular junction. Okay, this is the acetylcholine vesicles in which in this vesicle acetylcholine is stored up. Okay, this is your motor end plate. Botulism toxin is what it is doing is that it is blocking this particular NMJ junction, in which it is stopping the vesicles, inhibiting the vesicles from the release of this acetylcholine into the neuromuscular junction. Okay, so here you can see that botulism toxin is a zinc protease that hydrolyzes the fusion proteins required for exocytosis. So since it is hydrolyzing this fusion protein, exocytosis of this vesicle will not take place of the neurotransmitter acetylcholine release, preventing its release and leading to paralysis. As a result of which, this muscle fiber it undergoes paralysis. So this toxin will block a release of this acetylcholine from the vesicle. Stimulation is blocked. This is the thing which you have to remember from exam point of view. Then let's see why other options are not right. Option A was blocking sodium channels along the motor nerve. Sodium channels blockage is not in takes place. This particular thing is related to only acetylcholine. Where the sodium channels will come into picture, I will show you next slide. Inhibiting the reuptake of choline released, resulting in acetylcholine depletion. Since the acetylcholine itself has not been released into this neuromuscular junction, so there is no question of uptake of choline. Evoking unstimulated acetylcholine release, no, that is also not because we are stopping the acetylcholine release only from the vesicles. So these are the thing. Botulism is the main thing which is caused by this botulism toxin, and these are the other pictorial representation. This is the bacteria which is causing this botulism toxin. So in this video, what I try to do is that whatever the information with respect to botulism toxin is there, no, in the Clayson textbook, I try to collate all the information, all the pages which are mentioned in the Clayson textbook in this particular video. So first you read page number 88. Here in the regarding botulism toxin, it is given skeletal muscle paralysis caused by botulism toxin is due to inhibition of acetylcholine release from the motor neurons to prevent stimulation of this acetylcholine receptor at the NMJ junction. So you can see this receptor 1 in figure 3.17. This is the figure which is given in the page number 88 and is asking us to please go through receptor 1. This is the first option I was saying, the sodium channel blockade. Here the acetylcholine receptor it is there. Okay, this is the another pictorial representation how the synaptic place this acetylcholine release it is being stopped. Okay, along with this particular receptor 1, also read other type of things like voltage gated ion channels, excited receptors, exocytosis also in this particular figure 317. We'll go to next slide. So regarding botulism, I also found some information on page number 89. So please read table 3-4. In this they have given agents acting on signaling systems for neurotransmitters. Here we have acetylcholine nicotinic receptor. Okay, the location is skeletal muscle. Here the type of agent which is acting as agonist, and here agent which is acting as antagonist. Here under this we have nicotine, anatoxin A, cysteine and cholinesterase inhibitor. So our botulism will come under this cholinesterase inhibitor, and the effect is this: either nicotine, anatoxin A, cysteine will cause muscle fibrillation and then paralysis. Coming to agent tuberculin, lophotoxin from this CVIP, alpha bungarotoxin banded crate venom, alpha cobrotoxin from banded crate cobra venom, then erbotoxin B from branded crate venom, and botulinum. All these things, what they will do, they cause muscle paralysis. This you have to remember. Lophotoxin, CVIP, alpha conotoxin marine cone snail, alpha bungarotoxin, ergotoxin from batted gate venum, and alpha cobratoxin from cobra venum. Okay. In addition, on page number 1290, I have found, see, there is one list of LD50 values with respect to oral route. In that list of 10 to 12 chemicals will be there. In that last one, most potent one will be botulinum toxin, where LD50 value by oral route will be 0.00001 mg per kg. But here you can see with respect to intravenous route in the mice, the LD50 is 0.003 microgram per kg. So this is another important information that you can read about botulinum toxin on page number 1290. Let's also see what else is given in the page number 225. Here on Jan 16th, 2017, Botox made the cover of Time magazine under the title How Botox Became the Drug That Treating Everything: Depression, Heart Trouble, Migraine, Erectile Dysfunction, Back Pain, Sweaty Palms. So, almost all the problems. This was a publication which was there in the Times magazine. So, Botox has a large therapeutic protein. Here you can see about 1,300 amino acids derived from the botulinum neurotoxin type. Okay, A, which ranks as one of the most toxic substances known. So one of the most at a very low dose causing this toxicity comes the botulinum toxin, and the cause of the botulism following infection with the bacterium botulinum clostridium. So this is the thing you have to remember. So this manufacturer, no, Allergen, he has around 800 patents on this particular therapeutic application. So we should remember that Botox is probably the best known therapeutic protein, although insulin is it surpasses the insulin. So here you have to remember another thing is that monoclonal antibodies collectively account 25% of drugs approved by FDA. Okay, see, but rather than the conventional type of molecules, now we have therapeutic proteins, monoclonal antibodies, which also now become biologics are coming are slowly replacing the conventional type of drugs. You mean to say, therapeutic proteins as well as peptides are used and can potentially be used to treat infections, cancer, diabetes, cardiovascular diseases. Even Botox alone can be used for these particular conditions to the list. Okay. So let's see what else has been given on page number 1336 about botulinum toxin. Here there is another important thing: food safety through hazard analysis. Critical control point HACCP. This is the thing. It is given hazard analysis critical control endpoints. This HACCP is a management system in which food safety is addressed through the analysis and control of biological, chemical, and physical hazards from the raw material production, procurement, handling to manufacturing, distribution, and consumption of this finished product. The current practice of this HACCP was developed over time, but initial trigger came from the need to provide safe food. Initially, it was for only restricted to space as well as to the air flights, but later on it has been extended to other type of things. In the United States, the initial participants were United States Army Laboratories, National Aeronautics and Space Administration (NASA). Required contractors to identify critical control points. This is what we have: critical control points (CCP). Okay, it is the critical failure areas and eliminate them from the system. And also you have to remember about 1970, several cases of this Clostridium botulinum contaminations where because of this under processed low acid canned foods. In this food, no, at that time canned foods were there, in which the contamination of food with this Clostridium botulinum takes place, which by consuming this particular contaminated food, botulism will happen. The training program, Food Safety Through Hazard Analysis and Critical Control Point System, was held in 72, and it appears to the first published use of this term HACCP. So this also you have to remember one important point with respect to botulinum, which is given in Clayson textbook on page number 1336. Then we also have table 2710, which is given on page 1345. Here you see some foodborne bacterial exotoxins, not endotoxin, exotoxins and their mode of action. Botulinum toxin, which is a neurotoxin, and the producing organism is Clostridium botulinum. Toxicity of the host is it causes neurotoxicity, and the mode of action is it will interfere the normal transmission of nerve impulses by blocking neurotransmitter. What is that neurotransmitter? That is acetylcholine blockage. Acetylcholine is not being released into the synaptic cleft, released at the nerve terminals. Another important thing you have to remember is about lymph circulation. How lymph circulation and its connection with your botulinum toxin. Lymph circulation is important in the transfer of fats, large molecules such as botulinum toxin. We have seen one in the peptide which is having 1500 amino acids. We have seen say benzo. Other examples are also there. So lymph circulation is important. That is what is saying for the transfer of this botulinum toxin also, which is given on page number 1317. Next important last we have to learn from page number 1346. Some summary are given with respect to Clostridium botulinum. See, Clostridium botulinum is there, Clostridium botulicum, Clostridium barretti. These are food botulism. It is caused by eating food contaminated with botulinum toxin. Okay, canned food. Food associated with botulinum toxin include improperly canned low acid foods. What are those foods are green beans, corns, beets, asparagus, chili peppers, mushrooms, spinach, figs, baked potatoes, cheese sauce, beef stew, olives and tuna. Tuna fish canned also were there which are having this contaminated botulinum toxin. Toxin also may occur in smoked fish, fermented foods like seal flippers, salmon eggs also, and improperly home cured hams, meat, whatever this is. An increasing source of poisoning is from the use of flavored oils and oil infusion. Even oils are also having most typically garlic in oil preparations. All Clostridia, whether it is botulinum or Butyricum or Barretti, are gram-positive, spore-forming anaerobes, and they produce total of 7 serotypically and antigenically distinct botulinum neurotoxin. So what are the serotypes A to G are there? Out of which are more poisonous, more harmful that we'll see. Human botulism is caused by mainly by type A, type B, type E, and rarely by type F. So A, B, E, F are the things which are causing human botulism. C, D, and also E cause toxicity in other mammals, birds, and fish. Okay. Type G has not caused any, there is no reported, not caused any human illness. Okay, so the main mechanism once again last we'll see botulinum toxin is neurotoxic, interferes in neuronal transmission by blocking the release of acetylcholine at the peripheral nerve endings causing muscle paralysis. The end result is muscle paralysis. Toxin is elaborated in food wounds and infant gut. This is also very important. One question you might get about this also. Clinical illness is characterized by cranial nerve palsy followed by descending flaccid muscle paralysis, which can involve muscle of respiration. Okay, that is why artificial respiration need to be given in such cases. Recovery often takes weeks to months. Toxins are heat labile. This is again very important. Why they're able to replicate these particular strains of botulinum is that Clostridium strains are that because they are heat labile, may be rendered harmless at 80 degrees centigrade to 100 degrees centigrade for 5 to 10 minutes. Probably if the food is heated for 5 to 10 minutes at this high temperature, but the spores are among the most heat resistant. Even though the spores are resistant to this heat condition, they still survive. The case fatality rate in the United States is 4%. In the US, 4% mortality is there. Poison is fatal in 3 to 10 after have consumed this contaminated food. 3 to 10 days there might be a fatality case. Lethal dose is about 1 nanogram. So again I told you read about oral LD50, then IV intravenous LD50 in the case of mice, and also this 1 nanogram lethal dose. Whereas proteolytic stains called Clostridium botulinum group 1 may cause the food to appear. Another important thing is if you are unable to smell it and smell spoiled, if you are able to smell it, then at least you will not consume that food. May cause the food to appear and smell spoiled. Byproducts include isobutyric acid, isovaleric acid, and phenylpropionic acid. This is not the case with non-proteolytic strains. There are two types of strains, proteolytic strains and non-proteolytic strains. In case of non-proteolytic botulinum strains, which can flourish and elaborate toxins at temperature as low at very low temperatures, even at 3 degrees centigrade, these particular strains are able to survive. Current methods for detecting botulinum toxin include mouse assay as well as enzyme linked immunosorbent assay, with the mouse assay being accepted standard. The successful use of nitrates in meat, storing the meat by using nitrates to prevent spoilage by this Clostridium botulinum resulted in petition of FDA by the USDA to have sodium and potassium nitrate approved for use as prior sanction under 21 CFR 181.33. So these are the important points which were discussed, mentioned about botulinum toxin in the Clayton textbook. I have listed out all the pages as well as the tables. So all the information will be there in one video only. So any question with respect to this particular botulinum toxin, I will be able to answer in the exam. Thank you so much. Be curious. For any questions, you can always write a mail to arya.priyanka18@gmail.com. Like, subscribe, and follow my YouTube channel, Toxicology on Board. All the best for your exam.